I still don't see Destiny.
Oh, well, you said her name and she's here now.
Oh, yay.
All right,
Yep.
guys. It is 7:05. Well, 7:05 Central Central Daylight Time. So we're gonna get started. So good evening, good morning, wherever you are. Good afternoon. We're gonna get started with today's Pre-Med Peers Biology Class 2. I'm Nikhil, and today I have Karen with me. And let's get started. So who are we? We're Pre-Med Peers. We're a nonprofit in the Commonwealth State of Virginia. And you can always contact us through Slack, email, or Teams. And I am recording the session, or we are recording the session. So you can always refer back to the session and the slides that you'll get the access to shortly. So if you guys don't get everything down, don't worry too much. Feel free to email us and chat, send us your email through the chat so that we can add you to the newsletter or the listserv if you aren't added yet. Any questions so far? Also, please let me know that I've been speaking to you and not to myself for the past 2 minutes.
Yeah, no question.
All right, perfect. So today's agenda, we're going to start with a little bit of homework review from Kaplan's biology book, chapter 1. Just a couple questions that we've handpicked to make sure that we've got the content down. Then we're going to move on to some warm-up questions that we've designed for you, and then some content review about viruses, bacteria, and prions, and finally, a passage from Khan Academy that you guys can practice. Any questions about that?
Nope.
No.
All right, perfect. So if everyone can pull up their questions or the answers to their questions for chapter 1, that would be greatly appreciated. And please let me know if anyone would like to take a chance at answering question 1 for the end of chapter questions.
I can answer 1.
Yep, go ahead.
I said D.
And would you like to explain your answer choice?
Yeah, I can.
That'd be great.
I just have to fill up because I had the answer in front of me. Let me just.
No worries, no worries. If you'd like, I can pull that up as well, just so that we can all take a look at the questions.
Where can we find the questions in the homework? Because I looked under the bio second week and there was nothing there.
Gotcha. Generally, the questions are the end of chapter questions for the chapter you're reading for that lesson. And normally we will announce it in the chat for Teams if it's going to be something different or something specific. Is that a good answer?
Yes, I'm pulling up. So should I pull up the Kaplan document?
Yes, that'd be great. If you could pull up the Kaplan biology book and go to the end of chapter 1. Can you guys still see my screen?
Yeah.
That'd be great. 1 second, because I guess it backed out of the biology book. But yes, would the person that answered D for the first question like to explain how they got to the answer? And if not, I can go ahead and do that.
Yeah, so I chose D because it said that the chamber was full of like oxygen. And assuming that that's gonna like kill the bacterial infection. And so it's probably an obligate anaerobic bacteria because it needs it's not able to like survive with oxygen. And so I assume that it's causing the bacteria to die since it can't survive in oxygen or needs like no oxygen places.
That's great reasoning, actually. Does anyone have a different answer that they would like to share? If not, I can go ahead and explain the answer choices and why D is the right answer. All right. So I think the way I went about it, the key thing to see there is that while oxygen is normally present around us in air, they've increased the partial pressure of oxygen. So they're increasing the amount that is in the air in this chamber. So if they're using the increased level of oxygen as a treatment, we're looking for something that doesn't necessarily like oxygen. So when we look at the answer choices, we see aerobic, which means it uses oxygen. So we know that A isn't the answer. Then we look at the first word for the other three answer choices. Facultative, or I hope I'm pronouncing that right, means that it can use oxygen when it needs to. Aerotolerant, on the other hand, means that it can tolerate living or surviving with oxygen, but it can't use it. And obligate has to be combined with the next word, anaerobe, which means this bacteria can't use oxygen and it can't survive well in areas with oxygen. So that's why D is the right answer. Does that make sense so far?
Yes, it does.
Perfect. All right. For the next question, we've got question number 3. Would anyone like to take a stab at this one? If not, I can go ahead and explain it. So for this question, we're just looking at the types of nucleic acid that we can find in a virus. So the key thing to know about viruses is that they're very diverse. There are tons of them and there are tons of different types of them. So viruses can use both RNA and DNA, and they can use either single-stranded copies of RNA that can be positive or negative sense, and they can use single-stranded and double-stranded DNA. So in this case, the answer would be D. Would anyone like me to explain that further?
I just have a question. Which book is that?
This is the Kaplan biology book.
And what page is that?
Let's see here. This is page 101.
In the physical textbook,
Yes,
it should be page 43.
Or 48, depending on which edition you have.
Yeah, I have the post 2015 version here. So.
Sorry, did you say the answer was D?
Yes.
So I guess I was curious, can single-stranded DNA exist?
Yes, but single-stranded DNA is also normally subject to things such as nucleases, which is why in the capsid of a virus, it's a very protected environment. That's what the capsid is there for. It's there to protect the genome of the virus until it gets where it needs to go.
Okay, I was just wondering structurally, like the structure of DNA, like the binding of the bases, the hydrogen bond.
Gotcha. Yes, single-stranded DNA is definitely possible in nature. It just normally is easily degraded compared to double-stranded DNA.
Oh, perfect. Thanks.
I have a question.
Yes.
For like retroviruses, how like they start off with RNA and then they have to change to, they use reverse transcription to make the DNA. So like, is that like single-stranded DNA like included in that, even though it like, I guess would become double-stranded and then.
So that's one thing that I'm going to get to. So I'm going to hold off on completely answering that till a bit later. But the one thing I would like to say is we're just considering what will exist within the capsid of the virus when it is separate from the bacteria or the eukaryotic cell that it's infecting. Does that sound okay for now?
Yeah. Thank you.
Yeah, no worries. If there are no more questions, I'm going to go ahead and move to the next question in the homework, which is going to be question 9. Anyone willing to take a stab at this one? All right.
I said C. Sorry.
No, you're good. You're good. You're good. Go ahead. Do you mind explaining?
Yeah, so I said C because I'm rereading the question.
Mm-hmm.
Okay, so like eukaryotes and prokaryotes have different subunit weights, and then so that kicks out A, and then like prokaryotes don't have a nucleus, so that doesn't work, and then they don't have a membrane-bound organelles either. So like in eukaryotes do, so that kicks out D. And I don't know the presence of a membrane. I think they all have a membrane on the outside, so it wouldn't like differentiate.
Yep, perfect. Yeah, all cells that we normally look at, eukaryotes, prokaryotes, archaea, have membranes on the outside surface of the cell. So that's something they all share. Eukaryotes and prokaryotes do differ in their ribosomal subunit weight and the total ribosomal weight. Eukaryotes also do have a nucleus, while prokaryotes have a nucleolus, which is a less defined piece within the cell that holds the genetic material. And one of the key things that defines a eukaryotic cell is the presence of membrane-bound organelles like chloroplasts and mitochondria. So now that we know that A, B, and D all have differences between prokaryotic and eukaryotic cells, we know that C is the answer. Any questions about that? All right, I'm gonna move ahead to number 11 then. Was anyone able to answer this one?
I can answer it.
Yeah, go ahead.
Can you answer number 10 for us, please?
Yeah, I can answer number 10, but 10 isn't directly relevant to today's lesson, just so you know. But 10 would actually be C, since microfilaments are made out of actin, not tubulin. Does that make sense? But yeah, would anyone like to take a shot at 11?
Yes, I picked answer C.
All right, perfect. Do you have some reasoning for picking that one?
Yeah, I looked at A and B and they're not correct.
Mhm.
And then D, I was like, I didn't really see it happening, and C, it seems like I read it somewhere where viruses do incorporate like herpes simplex into the DNA of the cell.
Gotcha. Yeah, that's good reasoning. To explain that a bit further, A and B aren't correct because they're actually switched in terms of the cycle that they're in. If a virus is remaining dormant, it's considered to be part of the lysogenic cycle, where it is spreading without any symptoms or any extracellular replication. And during an outbreak, the virus would actually be in the lytic cycle, where it is reproducing as fast as it can and ripping open the cell through cell lysis. D is interesting because tail sheets and tail fibers are generally considered to be part of bacteriophages. And we have here in the question that HSV enters the human body. And we can actually confirm C as the right answer by looking at the keywords being remains dormant in the nervous system. And what we know about dormancy is that it's generally representative of the lysogenic cycle. And in the lysogenic cycle, we have the genome of the virus integrating itself into the DNA of the cell. Does that sound good to everyone?
Yes, it does.
Perfect. All right. So would anyone like to take a shot at 12? All right, I think I can take this one then. So for resistance to antibiotics, we're looking at how a bacteria is able to avoid these antibiotics by increasing its genetic variability. And the key thing we have to consider for bacteria is that there is a mechanism that they can undergo called horizontal gene transfer, which is why they are able to adapt very quickly along with their very quick life cycle. So we are looking at, in this case, the methods of horizontal gene transfer, which would be conjugation, transduction, and transformation. And since we do see conjugation and transduction here, we know that those two definitely have to be ways that they can adapt and be more variable. And binary fission, on the other hand, is just the method by which they reproduce. So that doesn't necessarily increase variability because they're just replicating an exact clone of themselves. So in this case, the answer would be C, 2 and 3 only. Does that sound good?
Yes.
Perfect. So for 13, would anyone like to take a stab at this one? All right, in this case, I will take it. So for 13, the key words that really stand out is bacterial recombination, the fertility factor, and an example of a novel process like other phenotypic characteristics, like a novel protein. So normally we see recombination happen again through horizontal gene transfer, where bacteria end up spreading their genes to each other. And we know that the process of conjugation, a type of horizontal gene transfer, requires the fertility factor that allows them to form sex pili that allow bacteria to connect and transfer their genes. So we know that with that information, we know that conjugation can't occur in a population that lacks the fertility factor that allows them to form these sex pili. So we know that A is the answer for this one. And please let me know if you have any questions, but I'm just going to continue onwards. For 14.
Question.
Oh, what's up?
Yeah. So this is my first time coming. So before we come, we should read the chapter. So if we're like on biochemistry, week 3, we would read the biochemistry book, chapter 3.
Yeah.
Okay.
I would say that the number of the class isn't exactly correlated to the topics that are going to be, or the chapter that you're supposed to read. So I would definitely say it should be on the schedule. So I would suggest taking a look at that. Like, this is the second class and we're on chapter 1 for the biology book. And if you have any questions about exactly what topics are going to be covered or what chapter you should read, feel free to type it in the Teams, and one of us will get back to you as soon as we can.
Okay, thank you.
Yep. So for 14, we are looking at Alzheimer's, where we have a protein that has a beta-pleated sheet structure and precipitates to form plaques. The key words that stand out to me for this question is the structure and the precipitation of the protein to form plaques. So in this case, we're looking at the pathogens that it might be most similar to, and we know that bacteria don't really have this beta-pleated sheet structure and plaques as their main method of infecting and taking control of cells. So I would knock bacteria off right there. Same thing with the viruses and the varioids, because those are all viral particles. Prions are what really stand out to me here, just because prions are really misfolded proteins that generally do have a beta-pleated structure that makes them more prone to the improper folding and precipitation, and I guess generally disrupting the formation and function of other proteins. So prions would be the right answer for 14. For 15, we're looking at, oh, yes.
I have a question, but it's on 13.
Gotcha.
So for C and D, like I thought like transduction always means like infection with a bacteriophage. So is that not the case?
Transduction, yes, that is the case, that the bacteriophage is transferring bacterial genes by just how it becomes, I guess, how it leaves the cell, how it's released from the cell. So you're right in considering both of them as being very interesting because they're actually the same answer. Transduction is infection and transfer of bacterial genes through a bacteriophage.
Okay. Oh, so it's kind of like a trick question.
Yeah.
Type of, okay.
They wanted to get those 4 answer choices, so they decided to really split up one of them.
Gotcha.
So for 15, we're looking at the genomic content of a virus, and the information we're given is that the virus must get to the nucleus in order to produce viral proteins. Now that's a key part of understanding this question and getting to the answer, because looking at central dogma and where these steps are done in bacterial cells or eukaryotic cells, we know that RNA is translated into protein in the cytoplasm or at ribosomes. So the fact that the viral particle must get to the nucleus to make proteins means that it's a DNA molecule because it first has to be transcribed into RNA and then translated into protein. So the answer for 15 would be A. So with that covered, we're going to jump back into the PowerPoint for today and move on to our warm-up questions.
A quick question before we move on.
Yes.
Could you quickly explain the difference between the nucleus, nuclei, and nucleolus?
Ooh, okay. Nuclei based is the plural.
Maybe we don't need to know it to that detail, but I was just thinking back to that question of the single membrane and.
Gotcha. Yeah. Okay. So that's the key difference between eukaryotes and prokaryotes, in that eukaryotes have a distinct nucleus, and the way we really see that is through a membrane that surrounds the nucleus, and a prokaryotic cell with its nucleolus doesn't have a defined membrane for the nucleus, and it's more just like a general area where the genetic material, genomic material is spread out. On the other hand, eukaryotic cell have the nucleus, which has a membrane surrounding it that is part of the endomembrane system.
Okay, so the first thing for the prokaryotes, it would be a nucleolus.
Yes.
Okay, perfect.
All right, now on to the warm-up questions. I'm going to give you guys about a minute to answer this one, and feel free to unmute yourself and put it out there because we're trying to be as interactive as possible today. I guess I kind of spoiled that one. So for the first one, the answer is actually glycolipids. And that is because for a virus, the genetic material consists of nucleic acids or nucleotides in DNA or RNA. We see proteins in the capsid that protects the genetic material and makes sure it gets to the place it needs to go for proper transcription and translation, or transcription is a maybe, depending on if it's DNA or RNA, and phospholipids. So the key thing with phospholipids in viruses is that they are included in the envelope of a virus, and not all viruses have envelopes, which is why I specified an enveloped virus. Does that make sense to everyone?
Yes.
Perfect. And our second question today. I want to give you guys an actual minute to answer this question instead of giving it away for free. And if you have the answer, please feel free to unmute yourself and just put it out there. Okay, so I think a minute is about up. Does anyone want to share their reasoning and or answer to this warm-up question?
I was like in between some answers because like I was thinking
Mhm.
about A or C just because for reverse transcriptase you need RNA to change the DNA.
Mhm.
Thought about like it being RNA dependent.
Gotcha.
So I guess I was leaning more towards C.
And why? Why would it not be any of the others? If you can answer that, or if you want.
Yeah, that's what I was trying to debate because I just knew like okay, for reverse you need RNA. Like you
Mhm.
can't reverse transcript. You need RNA to change the DNA. So if you don't have any RNA, like would you utilize reverse transcriptase? Like I guess it's just my random. I guess it's not a straight answer, just like my thinking.
Mhm. That's completely fine. There's going to be questions that I've definitely interpreted differently on the MCAT than I'm sure that the people who actually wrote the question meant for the test taker to interpret. So that's 100% normal. I will say in this case, you're definitely on the right track. And if you had to pick, I'm sure you would have picked C, as you said, and that would be the right answer. And to clarify, I just want to say that the polymerase means that's what they make. So an RNA polymerase would make RNA, a DNA polymerase would make DNA. And the dependent part is telling you about what they use to make the RNA or DNA. So an RNA dependent polymerase would use RNA, and the DNA dependent polymerase would use DNA. So the key thing about between the four that lets you pick C is that reverse transcriptase is a DNA polymerase. So it goes backwards from RNA to DNA. So we know that it's got to be a DNA polymerase. Or this one is DNA polymerase, but you have to also consider that it's RNA dependent because it used RNA to make the DNA. So now we have to compare that to RNA replicase and see, okay, which ones are they similar in? So an RNA replicase is RNA dependent. So it also uses RNA, but it makes RNA. So it's an RNA polymerase. So they're both similar in that they're RNA dependent, but they differ in that one is an RNA polymerase and one is a DNA polymerase. So the answer in this case is C. All right.
Can I ask a quick question from number 2?
Yeah, what's up?
So for the RNA polymerase or DNA polymerase,
Mhm.
suppose if you have RNA to make something, you would use an RNA polymerase, correct?
It depends on the protein that you're talking about. So if you're talking about RNA replicase, you would use RNA to make RNA. If you're talking about reverse transcriptase, you would use RNA to make DNA.
Okay.
So it really depends on the protein that the virus needs or the eukaryotic or prokaryotic cell needs.
Okay, thank you.
All right, so now we have question 3, which Karen will take over for.
Hi, everyone. Okay, so we have question 3. We split up these questions and you'll see soon why. A colony of bacteria with unlimited resources would not experience which 2 phases of growth? So you should be thinking about a certain graph from chapter 1. I'll give you a minute to decide what answer.
I'll go with B.
Do you have any reasoning behind that?
As long as it doesn't have unlimited resources, I would say it's not gonna go the exponential growth. And if it can go through the exponential growth, it's not gonna go in this stationary phase.
Okay, that's a very good guess. Anybody have any other answers?
I'm not too sure between C and D, but I think that the death phase wouldn't happen because there's death usually because of limited resources. So there wouldn't be a death phase.
That portion I will say is right.
Yeah, I was thinking C too, because I don't think it'd be stationary either, because it wouldn't stop growing since the resources are unlimited. So I thought C.
All right, that is actually the correct answer. I pressed spacebar, but it's actually it's on Nikhil's side. Could you go back one? Okay, so let me go through the answers real quick. So we have 4 phases of growth. We'll go over it again in the review slides. The first phase is the lag phase, and that will always happen with a colony of bacteria getting to know kind of a new set of resources because it needs time to kind of acclimate to that new environment. The exponential phase is when it really starts to kick off, and that's the second phase, is when it starts to kick off and the bacteria starts to consume all these resources, which allows kind of the colony to exponentially grow. And with unlimited resources, ideally, those are the 2 phases that would actually only happen because as long as there's resources, the bacteria colony will grow and it'll keep growing. The third and fourth phases, stationary phase and death phase, these happen when there's a limited amount of resources. So stationary phase is the third phase, and that's when kind of the resources are starting to run out. Kind of think like, oh, like the apocalypse is happening, and so the growth of the colony starts to slow down, and kind of starts to stagnate and kind of plateau. And the death phase is when the resources like really run out and the colony starts to die off altogether. So that's the 4 phases. Okay, so warm-up question number 4. So we have a plant pathologist, and he went to check on his tomato experiments, and he noticed that some of his plants, they weren't growing very much. They're stunted in growth. Lab analysis demonstrated that a large number of RNA sequences were silenced, limiting gene expression. What is the structure of the particle responsible for this? So this is kind of an indirect question.
I think I have an idea.
Go ahead.
Is this referring to a repressor?
No, this isn't so this isn't related to kind of so the particle I'm actually talking about is a subviral particle. So we're not kind of dealing with that
Oh.
topic. Yeah, we actually discussed that topic last time for the last class.
Is it referring to a siRNA? A silent RNA, like the small, not the small snRNA, but the siRNA, which is a post-transcriptional silencer.
That's a really good point that you made. I should probably have made my question more specific in that case, because that is true. They do their role is to silence RNA um, uh, post after transcription. So the keyword I'll say again is subviral. So it's a very specific particle that is covered in the virus section. I'll give you a couple more seconds. Okay, so the particle that I was talking about is a viroid. So it's a subviral particle that attacks mainly plants, and its structure is short, and it's also circular, and it's single-stranded RNA. So that's the structure of a viroid. They target mainly plants, and their function is to attach to RNA sequences, and this in turn limits gene expression in plants, and that can manifest in a number of ways, including including stunted growth. So, does that make sense to everybody?
Yes.
Oh, I have a question. Sorry. Like I selected A, but in terms of the difference between, like, is it just because it's circular between A and B? Like, is that like in terms of reasoning to differentiate between the two?
Typically, viroids they're usually circular, so that's what would kind of rule it out. Yeah. Okay, you guys ready for the content review?
Yes.
Yes.
All right.
All right. In that case, I'm going to try to move a little quickly just because I do want to get you guys out as soon as you can because we all have our lives and we are a little bit behind at the moment. So we'll start with viral structure and function. So a key thing about viruses is that they are tiny, absolutely tiny. They're in like the nanometers of size, which is degrees smaller than any eukaryotic or prokaryotic cell, and also helps them infect these cells. So the key ingredients of a virus is that we have to consider that there are no organelles or a nucleus because they are that small. And so everything is super simple. We have a capsid, which is a protein coat that can come in one of two general shapes, as we see at the top right. We see the helical shape all the way over at the top right, and we see the icosahedral shape. And this is a protein coat that protects the genomic material for the virus, and it's like a vehicle that carries it to where it needs to go within the cell that it infects. Then we have the genes, which, as I mentioned earlier in the warm-up, it can be almost anything in terms of nucleotides. It can be DNA or RNA, and it can be double-stranded or single-stranded, and it can be linear or circular. So it's pretty varied in that way. We've also got the envelope, which doesn't necessarily appear on every single virus. As you can see at the bottom right with the bacteriophage T4, we don't have an envelope, but an envelope, if it's there, is made out of lipids and proteins. Just like the cell membrane of a bacterial or eukaryotic cell. And there's a reason for that. sometimes these viruses, as you'll see later, pull off the cell membrane from these cells when they are released from the cell. And a key thing to remember about these envelopes is, just like other cells, they're sensitive to heat, detergents, and moisture, which is how we can break these viruses down, hopefully, for the most part. A key thing to remember about viruses is that they're all obligate parasites, which means they have to use some other living cell. Well, they're not technically considered living, but they have to use some cell and take control of that cell's machinery to reproduce and get the necessary things made that it needs to infect other cells. And I wanted to give you guys an example with the T4 bacteriophage, which is a great example of a bacteriophage. To explain a little bit further, a bacteriophage is a virus that infects bacteria. And as you can see with the T4 virus, it has some very special morphology to help it do so. It's got the sheath, which is this little cell that acts kind of like a spring, kind of like a syringe. The tail fibers, which the end of the tail fibers, since they're all protein, will have specific affinity for some receptors on some cells. And so these fibers will attach to the bacteria and orient it so that the base plate can align with the cell membrane. And pretty much what happens is there are some spikes here that stab into the bacterial cell membrane or cell wall, and the sheath compresses and releases the genetic material that is held inside the bacteriophage head. And this entire piece of the bacteriophage stays out while only the genetic material makes it in terms of a bacteriophage. And always, as always, let me know if you have any questions, but I'm going to keep going. So a little bit deeper into viral genomes. So these viral genomes can, as I said, be almost anything. It can be DNA, linear, circular, or single-stranded and double-stranded. The thing we have to consider here is that for RNA, there's a little bit of a difference in its life cycle based on if it is single-stranded and if it's positive or negative sense RNA. And the type of genome that it has a great effect on the size of the virus. So we have massive proteins like influenza, which if you see the picture at the bottom right, there are actually different strands of genetic material of DNA that can recombine. And that's why you see so many different types of flu. If you see the H1N1 flu, that's because the H1 and is a protein held on this strand of DNA that recombines, and the N1 is a specific recombination of these DNA strands as well. So the thing about this plus sense and negative sense RNA is that the positive sense is like mRNA for a eukaryotic or prokaryotic cell. It can directly be translated within the host cell to the proteins that the virus needs. On the other hand, the minus sense doesn't really code for a functional protein. So what it's got to do is it's got to take an RNA replicase or the host cell machinery to recreate the positive sense RNA that can then be translated into protein. And that's a bit of a longer step. And these two strands, the plus and the minus sense, the positive and the negative sense RNA strands, are always complementary. And so just a little bit further is that the plus sense is pretty much the same as an mRNA in a host cell, and it is directly translated to protein in the host cell. And for viruses that have a plus sense RNA in their genome, what we see is we first see translation of RNA replication proteins. So we have these RNA replication proteins that create more copies of the plus sense RNA so that you'll have multiple copies of genetic material to then translate into all the proteins that you need. This is just an efficient way of making the reproduction of viruses faster, since the more copies of genetic material you have, the more you can use at once to translate into protein. And in this case, that uses RNA-dependent RNA polymerase to recreate this RNA. Then we also have the negative sense RNA, which is a lot like the template strand of DNA, in that it isn't necessarily coding for a protein by itself, but it has the exact complement to the RNA strand that does. And what's necessary for viruses that hold negative sense RNA strands is RNA replicase. This is something that needs to be packaged in with the negative sense RNA into the virus. So when it infects a cell, it can automatically create the positive sense RNA. So that's really important in a negative sense RNA virus, to have this RNA replicase so that you can create the positive sense strand, or the mRNA, that can then be translated into protein. And we see that here. We see it synthesize this complementary strand using RNA replicase, and then you can use one of them as a template to make more genetic material to pack back into the virus once all the other proteins are created. And you can continue to use the negative strand to make these strands that can then be translated into proteins. Now, someone mentioned it before.
Go back really quick to the positive strand, the positive sense.
Yep.
RNA, okay.
Is there a specific question you wanted to ask, or are we just looking at the picture?
No, I just really wanted to know what was the difference between the two, like the major differences.
Between the positive sense RNA and the negative sense RNA?
Yes.
Gotcha. Okay. So as just RNA molecules, the only difference is that they're exact complements of each other. So they have the opposite bases, right? But when you consider them as part of a virus, positive sense RNA viruses have a little bit of an easier job because they can directly be translated to protein. So as soon as this RNA makes it into the cell, it just grabs hold of a ribosome and gets translated into protein. And the only other step it needs to make is it needs to get the RNA replicase so that it can make more of these RNAs to package into the new viruses. Is that good so far?
Yes, that's good.
Perfect. And then the negative sense viruses, on the other hand, since they don't directly code for protein, they need to be transferred or translated, I guess, translated into the positive sense version. So we're basically adding an extra step onto the plus sense RNA viruses, in that you need to make the plus sense RNA first before you can create the proteins involved. And that's why you need this RNA, because the host cell won't necessarily have an RNA replicase to make the plus sense strand. And that's why it needs to have that packaged in the virion itself.
I have a question. Since they're complementary to each other,
so
which one is which? Does it matter which one?
Well, obviously, well, no, I guess not obviously, but which one is which differs based on the virus and the protein you're trying to make, right? Only one of the RNAs will code for the protein that does the proper job that you're trying to do, and the other one will code for a random list of amino acids that isn't a functional protein, or honestly could just be like 1 or 2 amino acids if there's a stop codon.
So the positive one will always code for the mRNA that codes for the protein.
Well, the positive one is it's kind of like the same thing as an mRNA. So it just directly codes for the protein that the virus needs for its capsid or for RNA replication.
I see. Thank you.
Yeah.
So like in the bigger, so for the most like dangerous or effective viruses, do they tend to be positive sense since they don't have the extra like?
Well, as we'll see later, it's not necessarily the genome, but we have to consider the infectivity of the virus, right? Because adding an extra step doesn't necessarily make it a worse virus. It just, since it's already got this, I guess the ingredients it needs to do that extra step, it doesn't really add a difference outside of time. And even then, it's a slight time difference. We'd really have to look at the particles on the envelope of the virus or the method that it gets into the cell to look at how well the virus functions at infecting cells.
Gotcha. Thank you.
Mm-hmm. So we've gone through positive and negative sense RNA viruses, which are relatively simple compared to retroviruses. Retroviruses are a specific subsection of viruses, and they're all enveloped, and they're all single-stranded RNA. And usually you have 2 strands of this identical single-stranded RNA. The key thing about retroviruses is they use the envelope to combine with the cell membrane of the cell and release the capsid into the cytoplasm of the cell, as you can see at the bottom right. Now, the key thing here is that when the capsid releases the RNA, they use a protein called reverse transcriptase that takes them back to DNA, which can then be integrated into the genome of the cell. And in this case, once it's integrated into the genome of the cell, there really is no way to take it out unless you have a specific protein that looks for this foreign DNA and cuts it out, which really you can't recognize because it's using the exact same bases as every other DNA molecule in your genome. So really, the only way to kill a cell that is infected with a retrovirus that has made it to the genome is to kill the entire cell. And that's really important, which is why retroviruses are really, really, I guess, well known because of how good they are at doing this and how hard it is for us as humans or for bacteria to fight against this method. So we've got the reverse transcriptase, which is really important in helping these retroviruses evade our immune response because reverse transcriptase is really, really bad at what it does. It changes the RNA from the virus into DNA, but it makes tons of errors in doing so. And these errors are what help the virus stay variable in its genome and in the proteins it makes, so that the immune response we build won't necessarily work against the new mutated protein that it makes next. And that's super important. And these retroviruses, once they're integrated into the genome, will replicate within the genome of the cell when the cell replicates. And a great example of a retrovirus is HIV. And that error-prone transcription of reverse transcriptase is why HIV is so good at fighting off the drugs we give it and why we have to give bigger and different drugs every single time. So we've gone through the genomes, but how does the virus actually do what it does inside our cells? First, we have receptor binding, and that's the specificity of the particles on the envelope of the virus or of the virus's capsid or itself. And that means it can only infect a specific type of cell that has the receptor that binds to the protein on the virus. What happens after that is the receptor binding allows a sort of realignment, as you can see at the bottom right. Once the tail fibers bind to the receptors, it realigns so that it can inject the genome into the cell. And this is a bacteriophage, for example. So this realignment allows the fusion of the base plate to the cell membrane and the injection of genetic material. On the other hand, for enveloped viruses, what happens is, since the envelope and the cell membrane are super similar, the envelope will just merge into the cell membrane and release the capsid into the cell. And this, the release of the capsid into the cell, once the capsid breaks down, allows the nucleic material or genetic material, whether DNA or RNA, to replicate and to be transcribed in the case of DNA and translated into the proteins that it needs to make. And if we have DNA, it's going to make its way to the nucleus to be transcribed. And if it's RNA, it'll just replicate and be translated in the cytoplasm. So we see these RNA pieces use the host cell machinery to create both structural proteins for the capsid and for the other pieces of the virus, and we see it make viral genes, the replicate viral genes for the assembly of new viruses that can then infect even more cells. So, a key thing to remember here is that these new viruses are assembled by affinity. There isn't necessarily a machine or a protein that folds all these viruses together in the right orientation and the right shape. The evolutionary development of these viruses has enabled them to have an affinity so that when all the proteins are in the same generic area and all the DNA and RNA is in the same generic area, they just come together based on their affinity for each other. So how do they get out of the cell once they've replicated? So we have lysis, which happens when the cell notices that something wrong is going on, and it starts its own death processes like apoptosis, where the cell will break apart. And this breakdown of the cell basically acts like a balloon popping and releases all these new virions and viral particles out to the extracellular environment, where they can, in their case, hopefully go on and infect even more cells. Another way this can happen is through lysis, where there's just so many virions that the cell just bursts open and it releases, again, all its viruses to the extracellular environment. And then we also have extrusion, which is what happens in the case of enveloped viruses, where the virion, the capsid, and the genetic material fuse to the cell membrane. And this fusion to the cell membrane allows it to pull off a portion of the cell membrane with it when it gets released. And the translation of the RNA that has to do with the virus creates proteins that are relevant to it infecting new cells. And we can see that here, these RNA being translated into proteins that are then sent to the surface, and then the viruses attach to the cell membrane and pull it off there. And that's called extrusion. And the cycle by which this happens is called the productive cycle, because the cell is actively producing new viruses like a factory, but the cell is also left alive. And that brings me to the cycles by which a virus can infect and leave a cell, infect, replicate, and leave a cell. The lytic cycle, which is super simple, the bacteria, or the bacteriophage in this visual, or the virus in general, is just there to infect the cell, create as many copies as it can of itself, and get out as fast as possible. It disregards host survival completely. And viruses that are in this cycle are called virulent. Then on the other hand, we have the lysogenic cycle, which is a more behind the scenes approach. This is where the virus infects the cell, integrates itself into the cell's genomic material. So we have viral DNA that is now combined with bacteria or eukaryotic DNA, and then when the cell replicates, it also replicates this viral genetic material. Then when any sort of stressor happens that tells the virus, hey, I might not be able to survive as DNA in this cell for too long, what happens is a viral protein will then cut this viral genetic material out, reform it, and then it will translate it into the viral proteins and genetic material that are then formed into new viruses. So the three key steps here are integration into the genome, the stress trigger, which can be any sort of carcinogenic stress like radiation or some sort of chemical or even light that triggers it back into the lytic cycle. And this is a key way that bacterial transduction occurs, because when the viral DNA is cut out of the bacterial genome, it may accidentally bring along a couple bacterial genes with it. And then when the virus infects a new bacteria, those bacterial genes are then added to the new bacteria's genome. And that's how we see gene transfer between bacteria. The key thing about the lysogenic cycle is that there are some proteins that the virus will leave within the bacterial cell. And these proteins prevent other viruses from infecting the same bacteria. So they're like monitoring proteins. They monitor for any stress that the cell is going through, and they prevent the bacteria from being infected by any other virus, because they're trying to stake their claim on this cell to reproduce. And with that, we will move forward into subviral particles, where Karen will take over.
All right. Did you want to switch over the sharing? Or I can share it myself. Give us one moment, please. Okay. Can everybody see my screen?
Yep.
All right. So moving on. So Nikhil talked about everything viral almost. I'm going to be going over the last part, and then I'll be going into bacteria. So before we go into bacteria, we have the subviral particles. Sounds familiar? So there's two that we're going to discuss today. There's prions and viroids. So they're different in structure. They're different in what they target, and they're different in their function as well. So let's start with prions. So prions, they tend to be infectious proteins. So if you look at this picture here, it's got alpha helices, so it's indeed a protein. And its function is to trigger misfolding of other proteins. And that is not good when it comes to protein function. And so prions, they tend to target animals and humans. So I have some examples of diseases that are associated with prions. One of them is mad cow disease, which is with cows. I'm not going to go into that. And then insomnia with humans. So if you're having trouble sleeping, it might be a darn prion, man. And then we have viroids. These are small pathogens, and their structure is, as we went over in our homework question, or sorry, in our warm-up question, they are short and they're circular and they're single-stranded RNA. They target mainly plants, but there are exceptions. In the textbook that they mentioned that hepatitis B and hepatitis D are associated with viroids. So humans can be infected with viroids. And their function is to silence genes by attaching to RNA sequences, usually like large chunks of RNA sequences, and this prevents protein synthesis. So with prions, it disrupts protein expression. Viroids, it prevents proteins from even being made. So that's those two. And now going into bacteria. So bacteria is a prokaryote. And what mainly prokaryotes have these 5 things. If you look at the picture of bacteria, you can see that bacteria has a lot of other features. But I wanted to go over mainly the general prokaryotic structure that is general to prokaryotes and also bacteria. So we have the cell wall. If you look at the picture, it's kind of the little brownish layer in between the yellow and the capsule layer. And that protects, basically it's a protective layer. It's the main protective layer of the prokaryote. Bacteria, they have also a capsule wall, which allows them even more protection, but it protects kind of foreign things from getting in. And we'll go into this later. We'll go into this in the next slide as well. But there's two types of cell walls that you can see. There's gram-positive and gram-negative. I will explain that later. And then we have the cell membrane. An important thing to note, the MCAT likes to combine all sorts of things. You might see them combining like metabolism and cells. And in this case, one thing that you need to know is that the cell membrane contains the electron transport chain for prokaryotes. So compared to eukaryotes, they have the mitochondria, the powerhouse of the cell, very famous. But the prokaryotes, their electron transport chain is located directly on the cell membrane. Next, we have the nucleoid, which is different from a nucleus in that it's not the most sorry, there's less structure to it, basically. So in the regular nucleus in eukaryotes, you have kind of the nucleus, and then you have the nucleolus inside, which has the majority of kind of the DNA. In prokaryotes, you have just a nucleoid, which contains the DNA. So it's a lot less structured. And then next, we also have ribosomes, which are part of prokaryotes. Oh, my bad. Yeah. So it has ribosomes. If you notice here, so it just has ribosomes. There's not really any other compartment in the cell that is membrane bound. So anything like lysosomes, that's not in here. Anything like the Golgi apparatus, that's not in here. It's mainly just ribosomes that you'll see here. And lastly, it has the flagellum, which is like a tail. So you have the tail, and its main function is motility. So it helps the prokaryote travel through wherever it is. And specific to the bacteria, we have the capsule, we have the pilus, which is kind of like the little hairs here. And yeah, so moving on, we have the shapes. So there's three main shapes that we want you to know. I really don't know how to pronounce this first one. I think it's Kochi. I'll call it Kochi. I had a math teacher named Mr. Kochi. And these tend to be spherical. And so in this image here, you can see all the different types of spherical bacteria. It's not just one circle. It's usually, sometimes they can combine into multiple spheres. Next, we have bacilli, which is the bacteria shape associated with rod-shaped bacteria. So you have like your one that looks like a pill, you have one that looks like a bunch of pills sewn together, and you have one that looks kind of like, that's kind of a little more, that looks like that. I'm sorry, I'm a little tired. And then, so that's the bacilli, the rods. And lastly, we have spiruli, which tends to kind of be more spirally. And here's some examples of spirals. And if you have any questions, just let me know. And we'll also be taking questions at the end. So specifically going into the cell wall, we have, as I mentioned before, we have the gram-positive cell walls and we have the gram-negative cell walls. So there are a lot of differences between them. So when you're in the lab, gram-positive cell walls tend to be purple, while gram-negative cell walls tend to be pink. And structurally, gram-positive cell walls, they have a thick layer, of peptidoglycan. So in this picture here, it's kind of the purple strand here. And another thing that it has is lipoteichoic acid, or LPA. Together, the gram-positive cell walls, this structure is actually more effective at protecting the bacteria from triggering the immune response of the host that they're in. So this is technically having a lot of layers of peptidoglycan, and LPA is stronger against the immune response. And for gram-negative cell walls, it only has it doesn't have as much peptidoglycan, but it's still there. So so make a note of that. And then instead of more layers of peptidoglycan, it actually has an outer membrane, which is composed of phospholipids and lipopolysaccharides. And lipopolysaccharides specifically is more likely to trigger the immune response in the host. So it's a little more dangerous to be a gram-negative cell wall for bacteria if you're in your host. Next, we have bacterial classification by living conditions. So differentiating bacteria by by the environment that they're in. And mainly we're going to be focusing on how they react to oxygen. So, so the word aerobe, think oxygen. So we have these 4 test tubes, and notice that the bacteria noted by kind of like the red circles. So so Kochi bacteria, woohoo. And notice how these bacteria have different affinities to kind of the top. The reference point is really the top of the test tube, because that's where like the oxygen is basically. So you have this level here where the oxygen starts, and this first one here is obligate aerobes. So they need oxygen to survive. And so it's no surprise then that they're they're trying to get to the top of the test tube. They're like us, like when we're trying to come up for water, they need air. So that's why they're mainly at the top of the test tube. Obligate anaerobes, they want nothing to do with air, and they will probably die with it. So, or it will harm them. So they migrate as far as they can away from the top of the test tube or the oxygen, and they're stuck at the bottom here. So that's our obligate anaerobes. They are obligated to not have oxygen. They do not want it. Facultative anaerobes, they're kind of like wishy-washy. They sometimes need oxygen, they sometimes don't. So that's why you have a cluster here. They're like, oh, I need some oxygen. And then they're like, okay. And then some of them here are like, no, I'm good. I'm good on oxygen. I don't really need it. So it really depends on they switch on and off whether or not they need oxygen. And aerotolerant anaerobes, they're extremely neutral to oxygen. They don't use it technically, but they can survive in it. So they're kind of just chilling, basically. They don't really need it, but they can survive in an oxygen environment. Next, we have bacterial reproduction. Bacteria mainly reproduce through something called binary fission. It's a form of asexual reproduction. Asexual reproduction is mainly code for it reproduces by itself. As opposed to sexual reproduction, it requires interaction with a separate cell. So you have your cell here, and you have this prokaryote, this bacteria here. It's ready to replicate. And so compared to eukaryotic replication, this is, or like mitosis, something like mitosis, this is a lot simpler. There's a lot of steps skipped. So all you have to do is it just has to replicate the DNA, and so it's like the S phase, and then it just sorry, I'm like accidentally clicking. And then it just goes straight to telekinesis. So it starts forming its new wall, to its new cross wall. This is called invagination. And then once that cross wall forms, that's it. They got two new cells, and it has like the same DNA. So it's like they're like clones of themselves. They're like clones of each other. And because there's like the way I like to think of it is because there's like so few steps, it makes it like the speed is very fast. They replicate very fast, so faster than mitosis. And in the textbook, they said E. coli replicates in like 20 minutes. So like if that gets on your food and it's been a couple hours, just throw the whole thing away, right? And then next we have bacterial growth. So going back to our other warm-up question, like I said before, bacterial growth is dependent on on the resources available and the amount of resources available. So you have your bacterial growth curve. One thing to note is that this is actually the log form of the numbers of bacteria. So they kind of simplified this graph for you, but it's supposed to be like there's supposed to be a lot more curve to it. So instead of straight lines, they're actually they're actually a lot really dramatic curves, but they simplified it out here for you guys. So the first phase, we have the lag phase. So the bacteria, I'll go over it again. So we have the lag phase where the bacteria, they're at their new set of resources, they're acclimating, so they're kind of just getting used to the environment, and they don't really have time to grow when they're trying to get used to that. But once they have gotten used to it, they move forward into the exponential phase, or also known as the log phase, where their colony starts to increase exponentially. They start to divide at an divide at an exponential rate. And theoretically, it would just keep going and going and going. So if there were unlimited resources, it would just keep growing and growing and growing and just be exponential forever. But since in real life, there's always a limited amount of resources, so it moves into the stationary phase, when it's kind of when they're starting to use up all the resources, and their colony doesn't start to grow as much, and they start to plateau. And then as they start running out of resources, the colony starts to die off because there's no resources to keep them going, and the population of the colony plummets until there's none, and that's the death phase. So moving on, we have bacterial recombination. So how do bacteria transfer DNA to one another? And there's three that we're going to talk about today. We have conjugation, we have transformation, and we have transduction. So I'm going to start with conjugation here. We have two plasmids here, we have two bacteria here. One has a little sticky thing called a pilus, and one does not. The one with the pilus has something called a fertility factor, which allows it to basically has the power to pass on to pass on this pilus to other people. And so since this this would be known as an F positive cell, and then the one without the pilus is called an F negative cell because it does not have the fertility factor. And so step one, the F positive cell with the pilus forms a conjugation bridge with the F minus cell. So this when the pilus attaches to the F negative cell, this is called the conjugation bridge. And then from there, the F positive cell is able to transfer some of its DNA in one direction. It's transferring its gene to the F negative cell. So the F negative cell doesn't do anything, isn't transferring any genes to the other cell. So this is what we call unidirectional gene transfer. And bilateral gene transfer, it's the same as horizontal gene transfer. So it's the transfer of genes between cells. And while vertical gene transfer is typically within a cell. And then, so what can be transferred? Usually, sometimes it's usually DNA. And then most of the time, in this case, it's like the fertility factor. So it can transfer, F positive cell can transfer the fertility factor to the F negative cell. And so you can see in this fourth picture that now they both have the pilus, and so that means they're both F positive cells. The way I like to think of this is kind of like a vampire when the vampire bites you, you end up becoming a vampire too. So that's the way I like to think of it. The F positive cell can also transfer something called a plasmid, which is kind of extra genomic. So it's like it's not related to the genome, but it still contains information. And the plasmid can carry something called virulence factors, and virulence factors, this is a key word, make a note of this, you'll need this later. A virulence factor is anything that can increase the pathogenicity of a bacteria. So it makes it more pathogenic, so makes it a more dangerous pathogen. And when you have these virulence factors, and bacteria and when bacteria are able to pass it around to each other, this can increase like the antibiotic resistance among the said group. And antibiotic resistance is pretty self-explanatory. When you have an antibiotic, when you have a chemical that's designed to destroy the bacteria, antibiotic resistance means the bacteria has developed the ability to resist that antibiotic. It has the ability to resist being destroyed, and so that increases and so that's kind of what I mean by antibiotic resistance. And so that allows the colony in a lot of cases to get bigger. Next, we have transformation. So we have bacteria A, and then these are both bacteria B. So generally in transformation, there's a lot of ways that transformation can happen, but this is kind of the most general case. So bacteria A, it like lyses. So it pops out, it pops and it releases all of its contents, it spills its insides. And transformation happens when bacteria B ends up grabbing some of these contents from that bacteria A once had, and it incorporates it into its own genome. So that's kind of the main concept behind that. So you have lysing, and then you have the stealing, or you have like it obtains the outside genetic information, and then you have the integration. And then lastly, we have transduction. Transduction is different from conjugation and transformation in that it requires a vector. And most of the time, a vector is a vector. By vector, they mean that the bacteria can't do it on its own. So it needs an outside partner to help to aid it in incorporating new DNA from the outside. So typically it's through a virus. So you have like your virus here sitting on your bacteria. And the first step that happens is the infection of the bacteria. So you have the virus, it injects some of its DNA, and its DNA ends up becoming mixed with the bacterial DNA. From there, it's able to make, having the DNA inside the bacteria, it's able to make its own little baby viruses, so progeneration, so it's making progeny. So this is a lot like the lytic cycle. And so the progeny, they escape, the cell lyses, and then the progeny, they find a second bacteria. So these are two different bacteria here. Bacteria 1 is is one bacteria, and bacteriophore and bacteriophore is another one. So you have the new progeny, and it has both the host. It has both like its original DNA from the host, and it has the bacteria from the first bacteria. And what happens is, since this virus, this new progeny has both, it injects both of it into this brand new bacteria. And so now you have bacteria from bacteria 2 here, and then bacteria from bacteria 1 here, and then also some viral DNA. And then the two kinds of bacterial DNA are able to integrate into each other. So we have infection, step 1, integration, step 2, regeneration, step 3, the second infection into the second bacterial cell, and then integration into the second bacterial cell. So bacteria number 2 has DNA from both bacteria 1 and 2. So does anyone have any questions before we move on to the passage? Is anyone still here?
We're here.
Yes,
Awesome.
we're here.
Do you guys understand everything so far? Do you have any questions?
I have a question.
Sure.
Can we have the is there a PDF format of the slides? Do you keep it in a folder or no? Or do we have to go back to the recording?
You have the recording. Nikhil, we do share the slides, right? If I'm not correct, if I'm not wrong. Yeah.
I can send a link to these slides and allow you guys to view them. Let me copy the link.
That would be great. Thank you.
Yes.
Yeah, great question.
Yeah. Would we be able to get the slides before the class or no? Or would it be after?
It's going to end up being after. But for today's lesson, I'm going to post the link to today's slides in the chat right now. Hold on 1 second. Sorry, my computer almost died, so it's having a tough time working under.
Oh no.
This chat, or is it going to be on the Slack?
I can put it on both if you prefer the Slack. But yeah, I'm gonna send it in this chat right now, as soon as it will let me type. But for tonight, I don't think we're gonna get to cover the passage, but since we are sharing the link to the slides with you, it will have at the end of the passage, or at the end of the slides, it will have the link to the passage from Khan Academy and up to a PDF. And at the end of the slides, we do have the answers and explanations for each of the questions.
Yeah, so the passage is going to be just the PDF The PDF is just going to have the passage from Khan Academy, and then the questions without the answers. So you can do it on your own and kind of see what answer you would get on your own, and which is cool because Khan Academy usually have to guess right away. And then our answers will be, we have the answers, the screenshots to the answer, as well as the Khan Academy explanation on the slides.
Yeah, but
great, thank you.
Yep. And just can you guys get or access the link that I put in the chat?
I can't see the chat.
So on the top row, you have like a people emoji, and then you have like a speech bubble emoji. Click that one. That's the chat.
Oh,
Yeah,
it
I was
says
able
it's
to-
only available to team members.
Gotcha. All right, hold on 1 second.